A very good evening to all of you, and so today we are continuing with the previous topic. All set? Prepared for the next topic? So today's topic is comparatively simple than what we did in the last class, and especially if you have been through the last class, so it should be easy comparatively. Okay, so today we'll be talking about antigens, we'll be talking about antibody and antigen-antibody reactions. Now, only place where you'll feel, oh my god, what is this antigen-antibody reaction? Because we have a lot to remember there. We have a lot of lists, and it's important. You get MCQs there, and it's very, very important. Okay, so, but before that, yeah, things are easy. Other than that, we'll touch upon hypersensitivity today. Okay, so, good to start? Okay, so we start with antigen. Remember in the last class we were talking about these antigen presenting cells. They were eating up something and they were digesting it, putting it on a tray of MHCs and then taking it to the immune system, either, to the T cells rather. So now, what were they eating? What were the things they were actually phagocytosing? If these are antigen presenting cells, what is B? Is a foreign body, a pathogen, something which body is trying to defend. Good evening, Sana. Isn't it? So, now when actually the APC, antigen presenting cell, eats it and digests it, it makes, it takes out the gist out of it, and the antigenic part is presented to the immune system. Now, term antigen itself tells antigen. It is the part that helps the immune system generate the anti-soldiers, that is the antibody. Okay, so this is the part digested just out of those animals, which is or organisms or the foreign body, which is presented to the immune system. So what is antigen? Antigen is a substance which, whenever introduced parenterally, will stimulate the production of antibody. Now this is a natural way of going in, of antigen. But antigen can be introduced in the body intravenously or intramuscularly, intradermally, all those pathways. Basically, we can put it inside the body and body will respond to it by production of antibody. Now, antigen has to produce the antibody. It has to react with it. There are the two characteristics you can say. Okay. Now, based on that, we have some other terminology. We have a term that says hapten. Okay. Hapten is kind of an incomplete antigen, you can say. As I told you, antigen is to produce, it has to tell body to produce antibody. And after the antibody is produced, it has to react with it. Okay, these are the two things an antigen is doing. Now we have a thing that is called as hapten. Hapten is incapable of inducing antibodies. So it cannot tell the body to form the antibodies. But if there are pre-formed antibodies, it can react with them. So you can say something is missing in hapten. It is missing this particular part or property of antigen. So it is a kind of an incomplete antigen. Okay. So for a hapten to become complete, if you want to replace that missing property of hapten, we need to add this hapten to a carrier. So we plus carrier is equal to what? A complete antigen. Yes, perfect. Now, when we talk about the hapten, as we saw, they are not producing antibodies, but they are reacting with antibodies. When they react with antibodies, there are two possibilities again. They can, after reaction, precipitate with the antibodies, or the second thing, they can react, but whatever reaction is happening place is non-precipitating. The haptens that precipitate with the antibodies are called as complex hapten, while the haptens which do not precipitate with the antibody are called as simple hapten. So clear with antigens definition and haptens? Yes. Now we have an antigen. Supposedly this is an enlarged version of antigen that goes and stimulates the body to form antibody, and after that it will react with the antibody. Okay. Now supposedly this is the antibody that has been produced, and this antibody will now react with antigen. Okay, but when we talk about antigen reacting with antibody, it's not the whole molecule that comes, is responsible for forming this thing. So, the smallest part of antigen which actually brings about the production of antibody and reacts with it is called as epitope, while the same area on antibody that combines with that epitope is called as paratope. Okay, so epitope is present on the antigen and paratope is present on the antibody, and these are the corresponding regions which react with each other. Okay, so the smallest unit of antigenicity, the smallest unit that is required to bring about an antigenicity is your epitope, and the paratope will be the one combining with it to bring about the reaction. Think. You people can come up with a mnemonic probably. I can't think of any. Okay, whenever we have two things getting confused, or for example, we have two groups, some things to be divided in two groups, always tell your brain to remember only one. We all very well know that paratope and epitope are the corresponding things. If we try to remember as epitope on antigen, paratope on antibody, epitope on antigen, that becomes confusing. Okay, that's fair enough. E comes first and P comes later, and antigen comes first and antibody comes later, so that's fine. Paratope is antibody. Okay, fair enough. Nice one to remember. Anyways, after so much of discussion, you will not forget. So as I told, I'm bad in mnemonics. I can copy mnemonics from somewhere and tell you. So Rashmi, this will become my mnemonic forever now. Let's say come to antibodies. Okay, when we talk about the antibodies, okay guys, we are moving up to the antibodies. Okay, all of you gather up here. Now in antibodies, I will be covering a little bit more detail in the structure of antibody as compared to what would be given in your keynotes. So I want you all to have your notes and pen ready. My purpose is to explain the structure a little bit more in detail. Okay? So when we talk about antibodies, what are antibodies? Antibodies are chemically immunoglobulins. Now every time, yeah, that's okay, look at the class written on it. It should be fine. You won't even need the keynotes for now. Usually what happens, we cover totally in the class, and revision is best done with keynotes. So that's fine if you don't have the keynotes during the class. I'll tell you what else is the portion which is not there. Just for your information, there is the first diagram of antibody that will come up. of the structure. After that, whatever text slides are there that I have just added yesterday, so it's not there in your keynotes. But it's just to explain you the structure. Okay? So if you understand the structure, you don't even need to write it. It's all about understanding it. Okay? So when we talk about antibodies, okay, antibodies are chemically immunoglobulins. Right? Now, when we say chemically immunoglobulins, globulins means proteins. Okay? You all, when we go to actually biochemistry, you will know that proteins are of two varieties. One is the globular variety and one is the elongated version, what we call it, fibrous variety, fibrous proteins. So, globular proteins. So, when we talk about the globular proteins, here immunoglobulins. Immuno is because it is a part of the immune system. So, the protein which are a part of the immune system, and we call them immunoglobulins. There are some more characteristics to call them immunoglobulins. We need not go into those details, but just this is a chemical thing. Okay? So the immunoglobulin, that is the globular part of protein in the whole serum protein, makes up 20 to 25%. Okay? That doesn't mean that there are 20 to 25% of antibodies. Try to understand the difference. Okay, out of these immunoglobulins, some are antibodies. And there are some other immunoglobulins as well. Okay, that is the globular form of protein that take part in our immune reactions. Okay, antibodies are one of them. Right? That is why we say all antibodies are immunoglobulins, but not all immunoglobulins are antibodies. Whenever we have such a statement, right, it sounds like a tongue twister. But don't get confused. The term itself will tell you immunoglobulins is a chemical thing, chemical nature you can say of the proteins which act in immune system, out of which some are antibodies and some are something else. Okay? That's what you have to understand over here. Now, when we talk about immunoglobulin name, it tells us about that the protein is of globular nature and it functions in the immune system. Okay, but immunoglobulins, when we talk about the detailing of its chemical structure, they are glycoproteins. Okay, so they have these carbohydrates and protein moieties. So that's exactly its biochemical description when we say it glycoprotein. Okay, now when we, again, if I talk about a whole family of glycoproteins, immunoglobulins, just one kind of glycoproteins. There are some other kind of glycoproteins also which are not immunoglobulins, which are something else. Isn't it? It's something like if I say carbohydrates. It contains carbon and hydrogen. Same thing, when I say glycoproteins, they are the one having glycogen and proteins. Okay? And immunoglobulins are exactly, when we talk about the chemical structure, they are glycoproteins. I'm telling you this point very highlighted because, when we talk about the MCQs, you get immunoglobulin term used, you get glycoprotein term used, and you get antibody term used, and everybody uses them interchangeably. They're not interchangeable exactly. They're kind of, a part of something. One is part of other. Hopefully you understand. Clear or doubtful? Okay? Yeah, this is the diagram I was talking about. All the text slides after this I have added, recently. So, those are something which are not there in the keynotes. Okay? Now, when we talk about an antibody, give with me and don't leave it for later. We'll remember the structure now and today itself. Okay? So when we talk about an antibody, you'll see in this diagram, there are two light chains, these along the sides. Okay? And there are two heavy chains, the ones in the center. Okay? Now, if we actually use enzyme and try to split the antibodies molecule, we get two portions. One is FAB portion, this one, and the second is FC portion. Okay, This FAB portion is called FAB portion because it binds with antibody. Yes, antigen. Sorry, this is the portion that is antigen binding. Okay, of the antibody. That is why it is called as FAB portion. Okay, the other portion is responsible for other functions. It is a crystallizable fragment that is, chemically get crystallized. So it is called as FC portion. Right? Clear till now? Now, when we talk about the light chains, these two light chains along the side, these light chains are of only two varieties. They can be lambda or kappa. Okay, they are named after the investigators as whose names are Korngold and Lepari. Now, based on that, they have been just named as lambda and kappa. There is no significance other than that of their names. So, light chains are of two types, lambda and kappa. If you are having a habit of scribbling, it's very good, I tell you. Okay. Now, when we talk about the heavy chain, these two are heavy chains. They are of various varieties. Okay, and based on the different structures of heavy chains, your immunoglobulin, your antibody is classified into IG, G, A, M, D, and E. Okay? And the heavy chain which they have is called as gamma, alpha, mu, delta, and epsilon. Okay, these are the terms for the heavy chain associated with that. It's just corresponding with their name. G for gamma, A for alpha, that way. Okay, so we have 5 types of heavy chains. Clear till now? So you can say heavy chain is structurally different and antigenically distinct for each class of antibody. What are the classes of antibody? We have G, A, M, D, and E, and your heavy chain is different in each one of them. Right? Light chain of any one of them could be either lambda or kappa. Okay, so light chain is not the basis of classification. It's the heavy chain that is the basis of classification of the antibody. Right? Okay. Now, as I was talking about of separating these FAB and FC portion. Okay. No, long chain is not same for all, right? I told you, right? There are 5 varieties of long chain. And based on these 5 varieties, yeah, light chain. Sorry. Now, light chain is, similar in all of them. They can be in, for example, if you consider IgG, in IgG, we can have lambda or kappa. Light chain. There are only 2 varieties possible and it could be any one of them. Okay. It's the heavy chain that differentiates between G, A, M, and D. Similarly, A can have lambda or kappa. M can have lambda or kappa. But it doesn't matter. We are not classifying based on that. Okay, we are not considering it for classification. Fine? Okay. Now look at this division. When I say an enzyme used to divide the portion of an antibody into 2 parts, you have this FAB portion and you have this FC portion. Now look at the heavy chain. Heavy chain, it's here, it's here, it's here, and here. Isn't it? That means your FC portion is having nothing but the heavy chain. It doesn't have the light chain. Right? But your FAB portion has the heavy chain and light chain both. Okay. Clear? So, when I talk about this thing, this is heavy chain that is cut out, FC part of the heavy chain. Now, this whole is called as FAB. If I want to name the heavy chains of this FAB portion, we call it FDPs. Okay, make a note of this particular point. If you can just scribble and draw that particular diagram, it's as easy. It's very easy. It's from Anand. Okay. So, this portion of heavy chain is called as FDP. Clear? So when I say FAB, it will have both light chain and heavy chain. Out of that, the heavy chain part is called as FD. Okay. Now, when we see the whole molecule, okay, molecule of, oh, I'm sorry. When we see the whole molecule of antibody, okay, this whole molecule is made up of amino acids, okay, and one side, the terminal part is amino acid. The other side, we have a carboxy terminal, FD. Okay, so one side we have the amino terminus and the second side we have the carboxy terminus. So able to imagine where I'm talking about amino terminus and where I'm talking about carboxy terminus, and this thing is present in both the chains. Now look at this light chain. This is a light chain. Okay, I just colored it green now. This part of the light chain is amino terminus and this part of the light chain is carboxy terminus, because the light chain is terminating here itself, isn't it? So both light and heavy chain will have amino and carboxy terminus. The antigen combining site for the molecule is present on amino terminus, this side. Okay, now it is composed of both L and H chain. We already saw we have both H chain and L chain here in this particular region, and it is the amino terminus of both L and H chain. So it is an amino terminus of your whole antibody molecule, and that particular region is responsible for the antigen combining site, isn't it? Now, when we talk about that particular region being responsible for antigen, it's now this one will be easy to remember, amino terminus for antigen combining. Okay, A for it. Now, this particular thing, whenever we know that it has to combine with antigen, it has to differentiate between different kinds of antigen. Different kinds of antigen bring about different reactions of antibody. How can they do that? That means this particular region is variable. I mean, it has different structures and different, difference in the structure in different antibody molecules, and it is the one specifically that can bind with a different kind of antigen. So this is a variable region of antibody molecule. Does that make it clear? Does it sound clear? So amino terminus is the variable region. So even for the heavy chain this is the variable region, even for the light chain this is the variable region. Now let's talk about the light chain first. Now when we talk about the light chain is small. Okay, so approximately of 214 amino acid residues that make up the light chain, 107 constitute the carboxy terminus. That means this side. 107 make up the carboxy terminus. Okay. And this part is called as constant region. The other 107, now it's 214, right? So it's 107 amino terminus and 107 carboxy terminus. So you can say light chain is equally divided into variable region and constant region. Constant is carboxy, C for C. Okay? Amino acid sequence. So we are not talking about amino acid sequence as constant. Yes. Basically this is The constant region. Yes, the constant region is useful for many other biologic properties such as complement binding, and there are some general kind of biologic properties which an antibody will have. See, whenever an antigen will react with antibody, that one thing that antigen is reacting with antibody, that is one thing needed. After that reaction, this total thing has to bring about something else, isn't it? That is binding to some other known immunological cells or binding to the complement, and all those other functions are also to be done by the antibody. And most of the time that those other functions are responsible of that, the function of those constant region. Okay. Now when I talk about constant region of a light chain, we just saw that 204 is its amino acid composition. There is something called as FACS, which we don't have to bother about where it comes from. It's a light chain's composition. It will consist of 214 amino acid. Half is carboxy terminus and half is amino terminus. So 107, 107. Okay. Now when I talk about the light chain, this was our variable region, amino terminus, and this was our constant region, that is the carboxy terminus. The carboxy terminus part can have two types of sequences, kappa and lambda. Remember, our light chain could have been of two types, isn't it? Those two types are because of two different sequences that are possible in the constant region. Okay, the differences in the amino terminus or the variable region will actually result in its different antigen binding capacity. But difference in the constant region is giving it two types, that is kappa and lambda. Now amino terminus is variable, as we already saw. Okay, its variability decides the immunological specificity, that is, it is binding to which kind of antigen. Right? That is why this region is called as the variable region. In light chain, as we already saw, that the variable region is equal to constant region in size, or you can say number of amino acids. Clear? Hopefully you have a thing of, these light chain clear till now? Okay, good. Now let's come to the heavy chain. Okay, so this is a heavy chain. Okay, this was our light chain. Light chain had two regions. We already have it. Okay. Now H chain also has constant and variable regions. So we are again doing with the same concept. Now hopefully I don't need to explain what is constant and what is variable. You all know that. Okay. Now H chain's variable region constitute approximately only fifth of the chain. Now if I consider the whole chain, okay, this is the variable region of H chain. Okay, and this particular is the constant region of H chain, heavy chain, right? So the variable region is only a fifth, that is 1/5 of the total chain length, and it is located at its amino terminus. We already saw that variable is always towards the amino terminus, and yes, same is here. Only difference is your long chain is a long chain, but its variable region is comparatively smaller than the whole size of the chain. It just makes up 1/5 of the whole chain. Right? So if I ask you which region is longer in a heavy chain, it is the constant region. Okay? Now, when we talk about the variable region, that is that amino terminus side, these variable regions have some hypervariable spots. That means, now we all very well know that in variable region, the amino acid sequence is very variable. Okay, out of that, some particular region, the amino acid sequence is even more variable. Okay, that gives it fine control, you can say. Getting the point? So these hypervariable regions are called as hot the one mainly that form the antigen binding site. Okay. Now we all very well know that in antibody, a part of antibody that combines with epitope of the antigen was called as paratope. Isn't it? Now this paratope extends onto the variable and the hypervariable. Now it's obviously present in the amino terminus. That is a variable region. Now in the variable region there are some zone that is hypervariable, right? Now if you consider your paratope there, that is the region which is binding with your antigen. The part in the hypervariable region, the sites on the hypervariable region that makes actual contact with the epitope are called as complementary determining regions. They are the ones that actually, determine the interaction between the antigen and antibody. So they are called as PDRs, complementary determining regions. Okay, so they are present on hypervariable portions of the antibody. We are not saying, about light chain or heavy chain. We are considering either chains. Okay? Now, as put up by somebody before, when we talk about the carboxy terminus portion of the H chain, it does not possess the antigen combining activity, but it has other biological properties of the immunoglobulin molecules such as complement fixation, placental transfer, skin fixation, catabolic rate and all those things. These immunoglobulins have a life, they have to die, they have to, all those things are there, they have to do a lot of things, isn't it? So this is the carboxy terminus portion which determines all those things. Okay? Then now we saw about the two portions, that is we saw about the carboxy terminus and the amino terminus. Okay, we saw that there are two regions. Now these chains, how are these light chain and heavy chain joined together? They have intra-chain and inter-chain disulfide bonds. So there are disulfide bonds which are present inside the chain, okay, between these amino acid sequences, okay, and there are some disulfide linkages, these blue ones, that connect different chains. Getting the difference between intra-chain and inter-chain. Intra-chain is inside the chain, okay, so they are a part of the chain, right? Now these intra-chain disulfide bonds, they form loops. Okay, they form loops in the peptide chain. These loops are called as globular domain. Okay, and each of these globular domains will have a separate function. In the next slide, we'll actually see diagrammatically what functions they have. Guys, give me 2 minutes, I'll just be back, okay? Just revise in your brains what we just discussed. Try to draw a diagram so that you have the things clear what we have till now. I'll be back just in a minute. Okay, able to hear me? Yeah, so we were talking about these globular domains in the chains. Okay. I'll directly jump to the diagram to show you these globular domains. Now look at these things. Look at this diagram. There is a mistake actually in the Anant Narayan picture labeling. So if you are actually using, I think so it's 8th edition, just notice that mistake in the So, and because actually if you read the text, you will realize that mistake. So, it's not that big mistake, but you should be able to realize it. Okay? So, basically we were talking about these globular domains. See this intra-chain? They're forming these globular domains. Isn't it? Okay? Now, if you look at the light chain, this was a light chain. This is the variable region of the light chain. This is the constant region of the light chain. Isn't it? VL is nothing but the term used for the variable region of the light chain. You would actually have seen the MCQs with VL, VH and all that. Okay? So therein we are talking about the variable region of the light chain. VH is the variable region of the heavy chain. Okay? Now, in the constant region, we have these domains. Now, if you look at the light chain, there is only one domain. Okay? But if you look at the heavy chain, there are three domains. CH1, CH2 and CH3. Okay? Yeah, so they are the domains of the heavy chain. Between CH2 and CH1 here, okay, there is a hinge region. See that? Yes. So clear with the structure as of now? Okay, now we'll just go through the text and herein we have actually these hinge and all those things will have a lot of functions, but I'm just sticking onto the functions and details which are given in the book because we just expect from there, and rest of the other details you'll come to know as you go through a lot of medical MCQs. Okay, so as you go through medical MCQs, you'll come to know more details, and your basic understanding of the structure what we are dealing with today will help you easily understand those questions which are given in the medical side. But we have to sometimes restrict ourselves a lot as far as going deep into the theory because of the time constraint we have. Okay, but it's good at least to have the basics clear so that it's not a total mug-up scene. Right? Now CH2 region. Now when I say CH2 region, you should be able to immediately imagine where am I talking about. So CH2 region in IgG, it binds C1q in the classical complement sequence. So when you talk about the complement pathway, so this particular fragment in the complement pathway, it helps binding with CH2 region. So if it has to bind with IgG with that binds to the complement, it will be binding at CH2 region. CH3 domain, it mediates adherence with the monocyte surface. So are you able to imagine how these different domains are useful in different kind of functions of antibodies? All clear till now? And the hinge region that we were talking about. Okay, that was between CH1 and CH2. Okay, then comes the characteristics. Now this thing is there in your keynotes. Okay, now we have this IgG molecule. Now we all very well know that we have these 5 types. Now IgG has a half-life of 23 days. The percentage of Ig, total percentage among the all is 80%. So you can say it is the most abundant kind of immunoglobulin. It is distributed equally between intra and extravascular compartment. Now IgG is again divided into 4 types, IgG 1, 2, 3 and 4. And out of which the most abundant one is IgG 1. Okay? So keep having the things settled in. These are the fact-based information. Okay? Now look at this. It is a major serum immunoglobulin. Obviously it's the most highest component. It's the major serum immunoglobulin. Normal serum concentration is 8 to 16 mg per ml. Okay, you have a lot of MCQs from this chart. It crosses the placenta. Okay, understand this point because we'll be talking about this point's application as we go into the physiology. Okay, it crosses the placenta. It is not synthesized in fetus. Now, classical complement fixation is positive. Do you all remember at this point which CH region was responsible for binding of complement? CH2, and it was binding with C1Q. Okay, so look at this, because if you remember one point, obviously the other point will be, you'll be able to remember. It is the smallest immunoglobulin. It is most abundant, but it is the smallest immunoglobulin. It is also called as warm immunoglobulin. Okay, when we say warm immunoglobulins and cold immunoglobulins, basically warm immunoglobulins are the ones that interact with the antigen only at particular higher temperature, which are at the warmer temperature. Okay, they do not interact in the low temperature. Right? Now, catabolism varies with serum concentration. Forget about it. It's the predominant immunoglobulin in the gingival fluid. GCF. Now, this point is again important. Of course, you'll come across this again when we go on with perio, but you should be able to recall it that we already saw it in a chart in micro. Okay? So, not very difficult to remember. We'll move on with IgA. IgA has a half-life of 6 to 8 days. This is the fact. Look at this. IgG had 23 days. Here IgA has only 6 to 8 days. Okay? The percentage is 10 to 13. It's comparatively less in concentration. It's a major immunoglobulin in colostrum, saliva and tear, and this is an MCQ. You would have noticed an MCQ, this one. Isn't it? It's a secretory. Okay. Now it has two types, IgA1, IgA2. Okay. Now IgA1, IgA2, it lacks the disulfide bond between heavy and light chain. Okay. It is a minor component in serum, but it is a major component of secretory of immunoglobulin A. Now, what do I mean when I say secretory immunoglobulin A? Immunoglobulin A is present in serum definitely, but it is a major component of saliva, tears and all that where it is secreted. Okay? When it is secreted, it is a little bit in a different form, that is a secretory form. Yes. So, wherein it is called as secretory immunoglobulin A. The secretory form is a dimeric form, that is wherein there are two chains which are joined together by a chain. Okay, so secretory form is a dimeric form. Look at it here. Secretory immunoglobulin, many times written as SIGA. Okay, it is a dimeric form which is synthesized by the plasma cells joined by a J chain. This joining chain is called as J chain. Okay, and the serum thing is a monomeric form. Okay, this is, actually it is some cell-part unit. It's a unit to measure the length of an amino acid molecule, 7S molecule. When we say 7S molecule, I don't think so. Mucosal cells have an ability to synthesize immunoglobulin. They are a component of these things. They could be picked up from the inside through the serum and through pinocytosis released. I'll just check out on that today. I'll just check out on that part. It's a good question. If it says so, I need to actually, dig it more deeper. Where exactly it goes. Yeah, please do that. Okay. And yeah, there have been some questions I've been noticed. I think sort of yours. I on purpose didn't answer those questions because I was, we were going to come through these particular things, we were going to go through these particular things in the class. So I thought I'll leave that questions for you all. All of you can just refer those questions. They are good ones, of course. And, put some effort and put answers there. I will be watching that thread and I'll be commenting from my side as well. Right? So that will actually help you remember it more better. Okay? Yeah. And Uday, if you post your question also in the same thread, it will be better. We'll have that thread being quite valuable. We'll just bookmark. Okay. Now this Ig immunoglobulin A does not fix the complement. It can activate the alternate C pathway, but it does not fix the complement directly. Okay. Both IgA and IgG are secreted in milk. Now look at this point. This is again an MCQ. On fact based, okay. So don't just leave it for me. I'm reading it and trying to remember it. You also try to remember it with me. I'm gonna ask you in the next slide. Okay, IgA and IgG are secreted in milk. Which one was the one we, just now saw crosses placenta? There are only two we read, so it's easy. Okay, let's talk about IgM right now. Okay, IgM has a half-life of 5 days. अभी 5 पढ़ने के बाद बताना फिर मुझे आंसर. Okay, it makes up 5 to 10% of the total immunoglobulin family. 80% is intravascular in distribution. Now this point is very important. Now if you look at IgM, it stays in the blood. Most of the time it stays in the blood, and it is the one that protects against the antigens that are coming inside the blood. So the blood-borne infection main protection is by IgM immunoglobulins. Now again we have two varieties, IgM1 and IgM2. Now this is a very big molecule. That is why it is called as a millionaire molecule. It has a very heavy molecular weight. Now remember we were talking about IgA. It had 2 chains joined together by a J chain. IgM has 5. Actually, you people can open up the microbiology book and you will see this particular diagram. And there is again the J chain that joins these 5 molecules. Okay? So that J chain is also important, you can see. Okay, so it's a polymer of 5 joined by a J chain. It is the oldest immunoglobulin class and earliest immunoglobulin synthesized by fetus in response to infection. Okay? So, if by chance, immediately after the birth, if the fetus shows IgM, that means there was some infection inside. Why the fetus is synthesizing? Because IgM does not cross placenta. IgG crosses placenta. IgM does not cross placenta. It's very huge. Okay, and fetus synthesizes it after some weeks of birth. It doesn't synthesize it inside the womb. But if you see a fetus born and it has an IgM, that means it had an infection before. Okay? It is useful in diagnosing congenital infections such as syphilis, rubella, HIV, and all those things. Obviously, if there was a congenital infection, you will see an IgM antibody formed in the fetus as soon as it is born. They are short-lived and disappear earlier. That means there was an infection, IgM formed, okay, and disappear very soon. So if you see IgM, that means it's very recent infection. Okay, and they're just formed because if you see after some time, they'll be gone. Isn't it? So that's why they indicate recent infection. Okay, now here I'll be coming across a controversial statement. Okay, so just be with me and we have to deal with this MCQ very carefully. So it is 500 to 1000 times more effective than IgG in opsonization. Okay, but you have a question. Which of the following immunoglobulins are, which of the following act as opsonin? There is a question in general pathology that gives choice as IgG and C3b. It's not only IgG, it gives actually IgG and C3b. Actually, if you read Anant Narayan under opsonins, when it actually discusses opsonin, it just considers IgG and C3b. It doesn't talk much about IgM. Okay, but when you look in Anant Narayan, it tells that IgM is a very strong opsonin. There is a choice of Ig M in this particular question as well. Now, if you have IgG and C3b combination there, please understand that the question is coming from Harshmore. Okay, so going for that particular choice, you'll find a solid reference in there. Okay, if the question is in ComEd-K or KSET or some exam wherein you can claim, it's all fine, it's easy. But if it is in AVPG or some these kinds of exams, we may have to rely on matching the key. So many a times we have to try to match the key. Okay, so this will be one of the candidate questions for such kind of scene for you all. Okay, so I'm reading MCQ, remember all this. Yeah. So, yeah. Now, if you notice it is 100x times bactericidal, 20x time, it brings about the agglutination, but it is less effective against neutralization of viruses and toxins. Okay. Now, as we have already seen about the complements fixation, both IgM and IgG, they fix the complement, and it is a cold immunoglobulin. Where did we see warm immunoglobulin? Yes, both IgG and IgM are involved in type 2 cytotoxic reaction. When I say type 2 cytotoxic reaction, probably when we go to hypersensitivity, you'll be able to imagine more what am I talking about when I say type 2 hypersensitivity reactions. Okay. Then we have IgD. Okay, IgD, the life is 2 to 8 days. Can you remember the life of others at this point? IgG का कितना था? 93 नहीं, 23. Yeah. Okay, what was the next one? A का? What was A? 6 to 8. Hope you're not copying from the keynote. M was 5. Yeah. IgD is 2 to 8. Then IgE is 1 to 5. Okay? And again, IgD is intravascular. Which was the one which was intravascular previously we saw? Before IgD, which was the one? Yes. Now IgD again, we have two types, IgD1 and IgD2. The structure, it is similar to IgG. Okay? IgD and IgM serve as reorganizer receptors for antigen. Basically, they are the one present in the blood. Okay, they are the prime receptor of antigens in the blood, intravascular antigens. IgD is primarily responsible for activation of B lymphocyte for antibody production and suppression. Now, IgD is not that important. Its function goes in sync with MHG. Okay, so there's not much of the things which we need to know about it. IgE is then next one important one, which is again which is mostly extravascular. If you remember, this is the one which is mostly responsible for allergy and atopic reaction. Okay. So, if you notice, it has a unique property of heat lability, that is, it gets inactivated at 56 degrees centigrade. It is 56 degrees centigrade. Okay. It has affinity for the mast cells. It mediates this reaction. I'll be talking about this reaction. If you are making note, we can make a note of it. Actually, I'll be talking about this particular reaction where we use it. Okay. It has a role in anaphylaxis and parasitic infection. Don't go much into this IgE thing. It's just atopy, anaphylaxis, parasitic reactions, and probably its heat lability that may be important. Okay? So, try to recollect what I say for, I'll just end up this session here. In the next session, I'll start with a recap of what we just saw with immunoglobulins chart. Okay? Because that chart is important for you to know. For those who have the keynote, that's good. For those who do not have the keynotes. It's in Anant Naran. I'll leave. Okay? So you can just give a quick brush through. So see you all in the next part. So, bye for now. Okay. So videos there. Don't rush up to answer me these questions. I want the answers one after another as I ask. Okay? I know. Okay, answer to the first one. Which is the largest antibody? Answer to this one. Which are the antibodies which have J chain? Which is the antibody important in parasitic infection? Parasitic infection. It is IgE. Okay, which is the one responsible, which is the one antibody that crosses placenta? Is IgG a warm antibody or a cold antibody? Which is the antibody responsible for atopy? Which is a dimeric antibody? Is it only dimeric or it can be monomeric as well? If I say secretory immunoglobulin, it is dimeric. So if I ask you, which is a dimeric antibody, IgA would actually not be a right answer. It should be secretory IgA. That would be a right answer. Okay? Which is a millennial antibody? What is the half-life of M? Which is the antibody that is common in GCF? Which is the one that is responsible for intravascular? Yes, it's M and D, primarily M. The one that gives you the recent infection? Yes, it is